Hey everybody, today I'm going to present on cells, which is chapter 3 in the textbook, and it's the second topic in unit 1 out of 3 topics. In biology, you should have had some background on cells. I'm just going to quickly go through this, and you can look back at this video as many times as you need to. Also, I'll post a copy of this slide in the classroom. So cells are the smallest living unit of life. Of course, below that, you have atoms and molecules, but they're not living. They're not considered alive. So a cell is the smallest living unit, and it's composed of carbon, oxygen, hydrogen, and nitrogen. All cells have 3 main regions or parts. This is the nucleus, the cytoplasm, and the plasma membrane. The Hooke brothers is what I call them. You have Robert Hooke and Anton von Leeuwenhoek were some of the first to see the cell and see a living cell. So Robert saw a cell, which he was looking at a cork, which was not a living cell, but and then Anton von Leeuwenhoek was the first person to see a living cell, and he was looking at pond water. Next, in 1838, you had Slyden, and who concluded that cells make up all parts of a plant cell, and then Theodore Swan in 1839, just a year later, made the same claim about animals. Here are all the parts of an animal cell. Notice that you have ribosomes, and you also have peroxisomes, which you may not see in a plant cell. You have the nucleus, you have the cytoplasm, you have you're going to have the rough and smooth ERs. This is the mitochondria, Golgi apparatus, and some microvilli, peroxisomes, flagellum. In a plant cell, the main thing that sets it apart from an animal cell is your cell wall. You have a really thick cell wall, and you have a big central vacuole as well. Something different that's in plant cells and not in animal cells are chloroplasts. Inside chloroplasts, you have chlorophyll. Again, you have the nucleus and the cytoplasm as well. I want to also note that this is a permeable membrane, so things can come in and out of the cell of a plant cell as well as an animal cell. In the middle of the cell, you have the nucleus. This contains DNA. It's the control center of the brain. It has a membrane around it that lets things in and out. It's called the nuclear membrane. And then on the inside of the nucleus, you have a dark spot, which is the nucleolus. The nucleolus is where the DNA is found, and it's where ribosomes are assembled. Chromatin also is cell not dividing DNA forms. Chromatin is a conglomerate of pretty much of DNA, and so you have the cell is dividing, the DNA forms these chromosomes from the chromatin. The cell membrane is also known as the plasma membrane or the phospholipid bilayer. It has 2 jobs. The first one is to separate the cell from the outside environment, and the second job is for it to only let some things in and out. So it's that terminology for that is called selectively permeable. It consists of 2 phospholipid layers, which will have a tail. They'll be oriented tail to tail. The heads are hydrophilic and will be facing the inside of the cell and the outside of the cell. So it kind of looks something like this. You have this is called a phospholipid bilayer. Bi means 2. Phosphorus is going to be at the top, and you have a lipids on the inside. Here's another image of it where you'll have the phospholipid bilayer, and then inside that phospholipid bilayer you'll have proteins that were made in the ribosomes. Some of these protein channels are selectively permeable. Some of them are involved in active transport, and some of them are involved in inactive transport. In the cytoplasm, you have just cellular material. It's going to consist of anything that's outside the nucleus and inside the plasma membrane. So it's the site of most of the cellular activities. You're going to see an organelles there, such as the mitochondria, which consist of double membrane. It has inner folds, which are called cristae. So you see these inner folds are called cristae, and then also it carries out reactions in which oxygen is going to be used to break down foods and turn it in basically into a molecule called adenosine triphosphate, which is also known as ATP, and it is the main energy molecule in our bodies. It is the only energy molecule. In the cytoplasm, we also have the endoplasmic reticulum, also known as the ER, and it serves as a mini circulatory system for the cell. And it's also known as like the intracellular highway, so it moves things around the cell. The rough ER has ribosomes on them, which make it look rough. And ribosomes are the building materials of cells, and they are this is the site or the factory. This is where building materials, which are known as proteins, are made. You have micro proteins and macro proteins, and all the proteins in the body are made in the ribosomes. They're directed by DNA, and they are manufactured in ribosomes, and they are also sent into and out of the cell via the endoplasmic reticulum. This is what the rough ER would look like under a microscope, and then this is a smooth endoplasmic reticulum here. It wraps around the nucleus, so you have nucleus directing proteins to be made, and then they're going to be sent to other places. So the Golgi apparatus is just a stack of flattened membranous sacs associated with tiny vesicles. And these tiny vesicles' main function is to modify and package proteins. It's kind of like the post office. So at the post office, you take your package and they send it off to where it's supposed to go. And this is what's happening with the Golgi apparatus. The Golgi is just taking those proteins that were made in the ribosomes, and it's sending them to their place in the cell or outside of the cell. Inside the cell, you also have organelles known as lysosomes, and these are digestive. They have powerful digestive enzymes in them. So lyse means to cut, and lysosomes' main duty is to just digest worn out or non-usable cell structures and most foreign substances. You also have peroxisomes, which are organelles in the cytoplasm, and they contain oxidase enzymes that use oxygen to detoxify harmful and poisonous substances, including alcohol and formaldehyde. Most important function is to disarm free radicals, and free radicals are pretty much what kill you. So they're highly reactive chemicals that can scramble the structure of proteins and nucleic acids. So the peroxisomes will convert free radicals to hydrogen peroxide, and then catalase converts excess hydrogen peroxide to water. So there's numerous peroxisomes in liver cells and other cells, kidney cells. The cytoskeleton provides some internal framework that determines the cell shape. So the cytoskeleton is just supports other organelles in the shape of the cell. These are centrioles, and they can be found in the nucleus of the cell. They're known for generating microtubules, and these microtubules will basically pull or push the cell apart during cell division, and they direct the formation of mitotic spindles. We already talked about ribosomes. They are the site of protein synthesis, and they contain 2 subunits. They're made up of 2 subunits. Alright, that's all on part 1 of this video. I will post part 2 in another video. Thank you.